Thank you for taking the time to join us today. You won't be disappointed. We have two great speakers. What you're joining is the Human Health Exposure Analysis Research Exposomics webinar series. And the theme of this particular webinar, which is the sixth in the series, is systematic biological responses to environmental exposures. So in this webinar, we have two speakers, Dr. Baccarelli and Petrick, who will discuss how environmental exposures can affect such key biological mechanisms, including epigenetics, metabolic pathways, and gene regulatory pathways, and how these disturbances can result into human health-related problems. I don't want to take up too much more of this time, but I will say how today's webinar will be structured is each speaker has 20 minutes to give their presentation. We've got just about 4 minutes after each presentation for Q&A directed at the particular speaker, and then we'll have about 5 minutes for questions if you had for the other speaker or for both of the speakers. Also encourage you as questions are popping up in your mind, please go ahead and put them in the Q&A. And if by chance we don't have time to get to your question, we'll promise we'll circulate that question back to the presenter so that they could provide that answer for you in a follow-up email. So without further ado, I would like to get started. I'm going to keep the introduction very brief so we have as much time as possible for the presenters. And maybe I should just pause just for a moment to double check with Liz that I am coming through fine.
Yes, you are, Michelle.
Great, thank you so much. All right, so without further ado, let me introduce Dr. Andrea Baccarelli, who's our first speaker. He's a professor at the Mailman School of Public Health at the Columbia University in New York, and he is going to be presenting on omics, big data, and exposure sciences. So Andrea, I am going to turn it over to you.
Thank you so much for the invitation to be here today. Really a huge pleasure. And let me just make sure I can share my screen here. And
it's coming through we're looking at the right version.
Fantastic.
Seeing your slides.
Fantastic. And so I'm going to speak a little bit about what we do in my lab. But before doing that, I would like to stop a second and remind ourselves about the growing science and concept of the exposome, which we often define as the totality of exposures each of us experiences over our lifetime. And to contribute to this effort, my lab specifically uses molecular biology and data science to fingerprint chemical exposures. And specifically, we are lucky we can do that, not because we are great in our lab, but mostly because environmental exposures do leave lots of fingerprints on human cells. We can find fingerprints all over. In this review that Annette Peters, Tim Navrot, and I published last year, we classified these fingerprints over 8 different domains. And today I'll particularly be speaking about epigenetics and intercellular communication. Particularly, I would like to convince you that we can address 2 different challenges. So you will see 2 challenges that I prepare for us today and how epigenomics and particularly extracellular vesicles can address those challenges. So the first challenge is time. You heard me saying that the exposome is about the totality of the exposures we experience over our lifetime. And I would argue that it's very hard to capture the totality of exposure at one time, but it may even be harder to do it over extended time, especially with the ambitious goal of being the entire lifetime, all the moments of our life. So I would like to propose, though, a solution, which is epigenetics. Our cells record their memories in the epigenome. So the epigenome is a program that our cells use mostly to react to environmental cues. If the environment is great, we are all happy. If the environment is bad or adverse, the epigenome tries to help us cope with it. not only now, but it also embeds memories so that we are ready to react to the environment in the future. Particularly, this is accomplished via multiple mechanisms, but the mechanism we study in my lab is DNA methylation. DNA methylation is a program, and this is where the memories come from. Whenever we think about program, it is something that can be operational now, but it's also ready to be deployed in the future. So this is why it becomes memories. It's a program that controls genes. We have 20,000 genes in our cells, in our body, but usually each cell type, each cell actually only uses 1,000 of them. So only about 5% of our genes are on at any given time in one of our cells. And cells are amazing at that. They know exactly which genes to switch on and off at any time in our lives. And I'm always in awe when I think about our biology, how much our body does that we don't have to think about. And I already get in trouble to remember to pick up my coffee in the morning at the cafeteria. Think about if I had to think about switching on and off 1,000 genes at any time in my life. And these our cells do that automatically, and particularly they integrate the environment. Every time the environment comes at us, cells decide, take executive decisions, decide to, for instance, if air pollution is high today, they will switch on inflammatory genes and so on. And we have shown that quite a bit in our research. I would like to give you just one slide on the technology we use. We are using in our lab a lot of this microarray, which is a microarray produced by Illumina, which measures about 850,000 methylation sites. And this is where the second part of my presentation comes in, the data science, the big data. So for each DNA sample, and you might have noticed in my previous slide that there were drops of blood. We use a lot blood, mostly because it's the standard in clinical medicine, and it's easiest to collect, especially the easier source of DNA. We can measure for each blood sample, for each DNA sample, we get a readout, which is composed of 850,000 numbers. All of these numbers vary between 0 and 1, and particularly 0 means that there is no methylation across all the cells that we measure. 1 means that there is 100% methylation at that specific gene, that specific sites, 100% of the cells are methylated. And every number in between, of course, it's a range. So I want to show you now how using this rich data, we have shown that we can fingerprint environmental exposures. So the first paper is from a consortium led by Stephanie London at NIHS, to which we contributed. It's an interesting study because the results were really staggering. This is from the CHARGE consortium. It's a cohort of aging. We had about 16,000 people, each of them with DNA samples from 16 different cohorts. And we looked at smokers versus non-smokers. And what we found is that there were as many as 7,000 genes that had differences in DNA methylation. This is incredible because we had 20,000 genes, so about 40% of those were different. And that tells you how strong of an exposure tobacco smoking can be. But what was really interesting, and this is where I want to focus your attention, is that when we look at those who quit, former smokers, we found that most of these 7,000 genes went back to normal or non-smoker DNA methylation levels in a few years, some in a few months, some in a few years. But there were a small subset, which I'm showing here, that never seemed to go back to non-smoking levels. You can see here, particularly, it's a very interesting candidate, the aryl hydrocarbon receptor receptor repressor, which is part of the aryl hydrocarbon receptor pathway, which is a pathway known to be activated by PAHs, which are important component of tobacco smoke. And so you see here, this is a plot that shows the non-smoking levels. These are positive associations and negative associations. So you can see over the years, this subset never goes back to normal. So I used to often tell during my presentations that the idea I got from this work that we did together is that there are genes that are epigenetically annotated in pen, in ink, like these ones. Once they are marked, once they're programmed, these programs are almost impossible to erase. You can see after 30 plus years, they're still there, they still can be found. Some are instead annotated in pencil, perhaps some in hard pencil and some in soft pencil, in a way that we have a temporal dynamics where these methylation sites change at different rates. And in a way, this creates a situation where across these 850,000 methylation sites, we can almost leverage this data as it were a tape recorder that records everything or most of the things we are exposed to. And this, for instance, has been done with smoking. With smoking in particular, we can use machine learning to create predictors of smoking, estimates of smoking. There are a few that have been published over the past 3 or 4 years. These are all based on DNA methylation data. So on the type of data I just shown, they use elastic net machine learning to identify a few strong methylation sites, a few strong variables that can predict smoking, and then be used to create different predictors of smoking, whether someone is a smoker or not now, whether someone used to be a smoker, and also potentially the quantity of smoking, particularly pack years, which is a cumulative measure of smoking. And this has now become standards in estimating smoking. And I want to show you also that they are much better than self-reported smoking to predict smoking-related disease. So what I'm showing here is work by one of our postdocs who actually recently was promoted to assistant professor in pulmonology here at the medical school, Christina Eckhart. And she found that she used one of these methylation-based biomarkers in blood, epismoker, which is one of the simplest biomarkers. It just tells us whether someone is a smoker or not based on DNA methylation data, even without asking them. And she looked at lung function loss, chronic lung disease over time, and mortality. And you can see how the survival curves show huge separations. These are the people who are predicted to be smokers. These are the people who are non-smokers. And you can see the airflow limitation, chronic lung disease, and also all-cause mortality. This is something we know. Smoking does that to you. But what was really interesting is that when Christina compared this data based on DNA methylation, this analysis based on DNA methylation to the actual self-reported smoking, the DNA methylation was much associated in a way that was much more stronger to the disease and to mortality. So in a way, I can't say they are better biomarkers of exposure, but I can say, I want to say that they certainly are associated to smoking-related disease in a way that is much stronger. And perhaps because there are different types of biomarkers that are not just biomarker of whether people smoke or not, let alone the fact that there is error in self-reported smoking, but there are also biomarker that integrates whether the type of smoking patterns, what we call smoking topography, how much we inhale and exhale, and also whether the smoking products get into the lungs, into the blood, to the point of getting the cells, epigenome react to them. So it's a really interesting biomarker, perhaps closer to the target tissue. I also rapidly, I would like to tell you that we have used exactly the same approach to create blood DNA methylation biomarkers of lead. We did that using a special data set in the normative VA study in Boston, where we had measures of bone lead. As you know, lead accumulates in bones. It has a half-life of more than 10 years in bone. So we were able to train our machine learning onto this special data set, and we obtained a biomarker that reflects long-term lead exposure, which is something unique, a blood-based biomarker that doesn't exist so far. Blood-based biomarkers only record usually, or lead, only record a few months of exposure. Here we're talking about years of exposure. And also we have data that, according to your time, I'm not showing that this, please dream about the future, about 10 years, 20 years from now together. And one day we might walk into our doctor's office or are going to have cholesterol, lipids, glucose, but perhaps we will have by then even environmental blood tests, which is DNA methylation based. And you would receive a report like the one here, which I made up, of course, where they would say, you know, you are great, you have a great diet, you have a great diet over here. You have also, you are doing good with radiation, but you know, there are some issues with metals. Perhaps we need to test your water or see where this might be coming from. And also there's issues with air pollution, which of course is a societal problem. We can't bring the burden on individuals to clear air pollution. And down the line, all of this is affecting your risk of disease. This is not that far. I mean, we are already working toward this type of work. And honestly, I think we have invested a lot of time in using epigenetics as a mechanism, but perhaps it's going to be much more fruitful in the future as an outcome, as a profile, as a way to profile the environment, to fingerprint different exposures. In 5 minutes, I would like to talk about the second challenge, which is about tissues. If you switch on Siri and you ask Siri, hey, tell me how many cell types there are in the human body, Siri will tell you there are about 200 cell types. But when we do this type of analysis, we usually measure only one of them or a mix of them, like blood. So how do we get to measure exposures into the tissue of concern? And this is mostly motivated by work that my former postdoc Xu Gao and I have done on showing links between air pollution and cognitive decline and dementia, and where we found clearly consistent associations between living in areas with lower air quality and cognitive decline in older people. And of course, I mean, this is how do we measure exposures in the brain? It's inaccessible. No one wants to give out parts of their brains before it's time. And so really it's hard to measure the exposures in the cells, in the brain cells. So one solution that we have is extracellular vesicles. These are membrane-bound nanovesicles found in any human biofluids, including blood, plasma, or serum, which is a previously hidden signaling system. We think that they work a little bit like a new inflammatory system or a new endocrine system, just to take license on comparing to other things. But what is really important today is that we have been setting up in our lab methods to isolate in plasma vesicles that come from neurons, and these cross the blood-brain barrier, so they really can come from the brain. We do that because these vesicles have on the surface proteins that are the same as those found in neurons, so we can use antibody or flow sorting to isolate and enrich, actually, I should say, the sample, the preparation for neuron-derived vesicles. Specifically for this audience, I would like to highlight the work that my colleague Diane Ray has been doing here at Columbia about being able to measure chemicals. She's done a lot of work actually on metals that I should not call them chemicals, but potentially we can measure anything we want. And she was able to show that by measuring metals in neuron-derived EVs, she can produce a biomarker of brain metal load that is much more effective than measuring lead anywhere else, and metals in general. So, and our plan, we have been funded by an R35 from NIHS. Our grand plan is to create an atlas and make it publicly available, where we are going to create methods to isolate vesicles that come from 20 plus different cell types so that we can interrogate different tissues and cell types. Of course, there are a lot of challenges. First of all, I only showed examples about 2 or 3 exposures, but the number of exposures are countless and there is a lot of work to do. If we want really to build lifetime molecular biomarkers, we also need lifetime data of exposure. So in a way, smoking is a low-hanging fruit because you can ask them how long they've been smoking and when. It's much harder with other exposures. And of course, we need to have validation and implementation across different populations. So I would like at this point to stop here to thank everyone for their attention, especially I do very little at this point in my lab. My lab has been amazing in doing all of this. You saw some of the people, and thank you to the NIHS for the strong support in funding to my research. Thank you so much. I'll be happy to take any questions.
Thank you, Andrea. Just a reminder to participants, please pop your questions in the Q&A if you have any questions. And if you find yourself with a question later on, don't be shy to go ahead and put it in later on, and we'll be sure to get to it. I had a question for you. Earlier on, you were talking a little bit about that range. So a 0 to 1, no methylation to full methylation. And I'm wondering what your thoughts are in terms of looking at single cell analysis and whether you need to really dig into that to understand how patterns might change. One thing that comes to mind is, do you end up diluting the signal out or not if you're looking at more than one cell or even more than one cell type?
Yeah, I mean, it's a great idea and something we haven't yet delved in, but clearly single cell sequencing opens lots of opportunities to study things that we cannot do as an average. We're getting an average of all the cells that go by the scanner in the microarray or in the sequencing. We're starting to work with sequencing now. A single cell sequencing can tell us a lot more about the combination and the pattern of methylation within the same cell, which honestly would be a great data science exercise to use that combination, that fingerprint, rather than the average across cells. And also will help us to do more in identify which cells we are actually sampling, because white blood cells is a huge group of cells. It's not just one type. Yeah.
Yep, absolutely. So another question is the utility for looking at DNA methylation. Is this something that you could look at as a readout for many types of exposures, or is it a certain class of chemicals that are methylating DNA? Can you comment on that?
Absolutely. I mean, ideally, I would want to say that we can capture anything everywhere. We need to be aware that these are responses that our body builds up in reaction to mostly cues around us, and some cues are stronger than others. For instance, we clearly see that lead changes the methylome very strongly. Smoking, of course, changes very strongly. But when we looked at white blood cells, in particularly blood, we need to think that most likely these are changes that control and modulate inflammation and immunity. So we think we are probably going to have better luck for chemicals that affect inflammation and immunity, or in general white blood cells. We might be more limited in for other types of cells. But my ideal long-term objective would be to have one single test, and I want to remark one single test, one single measurement done at one time, where you could have an output with 2,000 exposures. But again, we might be limited in what we can query. And I have to say, this test is becoming cheap. I mean, I can see how this might be. The microarray might go down to a few tens of dollars per sample over time.
There's another question we have is regarding for the smoking epigenetic markers, were there follow-up on the functions on the specific genes?
Yeah, I mean, of course, not all of them, because to follow up 7,000 genes, it would take probably 7,000 of us. But this is the problem with epigenome-wide association study, or this type of discovery analysis, where really you get a lot of things that you never heard of. But in the gene that is most strongly associated with smoking is the aryl hydrocarbon receptor repressor. The function is really interesting. It's a gene, again, that gets activated in this case to shut down the receptor that is sensitive to smoking. So it really seems to be a modulation mechanism, perhaps a defense mechanism, that make our cells live in an environment that is smoking rich, to use a word that we are making up right now. So this is particularly interesting. By the way, at some point we gave an exercise in one of our boot camps where we introduced people to epigenetics here at Columbia, where we gave people just a data set with 10 smokers and 10 non-smokers, and really thought we would not see any statistical significance. But instead, the HHEAR, the aryl hydrocarbon receptor, was significantly different in methylation, even with a sample size of 20. Bonferroni corrected. So something we usually think we need thousands of samples. So this is so strong that even a sample, we did it with 16,000 people. Apparently, we needed only 20.
Okay, we need to move to the next presentation, but I am very intrigued by these extracellular vesicles. So I'm going to pop a question in, and hopefully we have time at the end. If not, I'm going to ask for you to send me the answer to that separately. All right, so thank you. So please hold on, because hopefully what we're hoping for is at the end we can do put in additional questions for Andrea, should anyone have any. All right, so next up we have Dr. Lauren Petrick, who comes to us as an associate professor from the Icahn School of Medicine out of Mount Sinai, New York. And she is going to give us a presentation that's entitled Metabolomics, Gaining Insights into Biological Responses After Exposures. So Lauren, I'm going to turn it over to you.
Great. So can you hear me?
Slide in. I sure can.
Great. Okay, so thank you for having me here today. I'm going to be discussing the role of biological response, and especially metabolomics, in the exposome concept. And so I'm going to be using mostly evidence provided from other investigators and a little bit of my own at the end to make the case that there's a link between the exposome and the metabolome, and that the field is starting to come up with strategies to address some key problems. And some of these include dealing with mixtures, since we know we're not exposed to single toxicants, but mixtures of chemicals, exposures that may be transient or chronic, and potentially at low concentration levels. So as we all know, exposomics research aims to investigate the health-related effects of exposures. And one technology that has been developed in the last decade is using mass spectrometry-based untargeted analysis to measure metabolite and chemical profiles in a biological fluid like blood, urine, or saliva. And so in this figure here, traditional metabolomics, sort of the dashed bottom over here, focuses on measures of endogenous small molecules as outputs of metabolic pathways, where changes in these sort of omic layers can lead to changes in metabolite profiles that are then linked with phenotypes. So exposomics, sort of this bluish above, expands on this and includes measurements of exogenous small molecules that then become circulating and can influence this omic cascade. So from an analytical perspective, there are two types of metabolomic studies. These are targeted versus untargeted. And a targeted assay is used when you know which metabolites you want to study and you measure those quantitatively, which makes it more easy to compare across studies and populations. In an untargeted metabolomics assay, we spread a wider net and make no assumptions on what is important in the study. And we measure 1,000 to 20,000 or so unknown features semi-quantitatively. And we do this because we want to generate new hypotheses on novel biomarkers of exposure, biological response, and phenotypes. So for an example of the power of a targeted metabolomics assay, this study shows how metabolite profiles can link biology to health outcomes. And here, targeted metabolomics data was combined from several studies to form really an impressive collection of 120,000 participants from the UK Biobank to evaluate whether metabolomic profiles can inform on multi-disease risk beyond conventional clinical predictors. And so they use neural networks to learn disease-specific metabolomic states, and then tested these in independent test sets for 24 common health outcomes. This included cardiovascular, diabetes, and cancer. So even though they were limited to only 170 metabolites and biomarkers, they found that the metabolomic states were predictive for all outcomes except for breast cancer. And of course, there was some overlap between the metabolomics information and the clinical predictors for most of the diseases. But there were 8 diseases in which the metabolomics markers added predictive ability. And so this figure demonstrates how metabolomics improves the prediction of type 2 diabetes. This is prediction 10 years after the baseline. And that's compared to using 3 different clinical predictors alone. And so the first clinical model here used just age and sex. The second used an algorithm that includes things like blood pressure and cholesterol. And then this third clinical panel was a broad-based panel that included laboratory clinical measures. And so you can see this goodness of fit here on the y-axis for the metabolomics algorithm is above 0.8 compared to this model of just age and sex, which is around 0.6. And then the green dot is when those two models were combined. And as we move across the x-axis, each time the clinical model included metabolomics, the prediction was improved. And this study demonstrates the ability of the metabolome to inform disease risk. So the question is, can the metabolome also inform on environmental exposures? And so current high-resolution mass spec approaches cannot distinguish the sources of the molecules that are present in these biological samples. They just measure what they detect. So measurements of the blood and urine exposome will overlap with the blood and urine metabolome. And analytically, an untargeted metabolomics assay will capture and measure environmental chemicals as well as endogenous metabolites. And so this is an example of the breadth of exogenous and endogenous metabolites identified using an LC-MS assay. And so on the bottom left, this here is a network of endogenous metabolites in red and exogenous in gray, measured in maternal blood collected from around 300 women during pregnancy. And then this subnetwork illustrates correlations between endogenous metabolites, exogenous compounds, and these include things like PFAS and parabens that are correlated with hormones and fatty acids and other endogenous metabolites. And so this study, as well as others, demonstrate the diversity of endogenous and exogenous exposures, which can be captured in these experiments. But determining associations between these chemicals and health in populations is still a challenge because exposures are highly dynamic and spatiotemporally different, right? So this is an example. I'm showing you a study that was performed in a group of people that were asked to walk down 2 different streets in London, Hyde Park and Oxford Street. And Oxford Street has a lot of high traffic related pollution, diesel vehicles, and this area of Hyde Park has very, very little traffic. And you can see that even within this city, the differences in levels of black carbon, NO2, and others are very different between the 2 locations. In addition, you can see that there's actually very large variability in the individual's exposures, even within the same street. And this is likely dependent on the time of day, related to traffic or weather patterns. And so bringing these external exposures into the internal blood measures, this is an example of a single individual where internal chemical exposures were measured over a 50-day period. And here are the 13 classes of chemicals that are listed here, and they varied in time according to these sort of circle plots up here, where the agrochemicals, these are the ones in green, showed a 29% increase and a 13% decrease across those time windows, depending on the individual chemical. So if you look over here at the heat map, you'll see this sort of sudden increase in concentrations where it moves from dark to light. And this occurred after January 25th, which coincided with the start of some travel of this individual to different geographic locations. So the chemical exposome was greatly impacted by the individual's spatial changes, even for a healthy individual over a short period of time. So in the same study, multi-omics was performed according to the schematic on the left. And this includes measures of the metabolome, the proteome, cytokines, clinical blood tests, and gut microbiome. And they looked for correlations between the set of measured chemical exposures, external environmental exposures like air pollution and temperature and humidity, and biological exposures as well from fungi and other organisms. And interestingly, they found that chemical exposures correlated the most with the metabolomic omic layered, and that's what's pictured here in green. The proportion of chemical exposures linked with the metabolome was the highest at 42% compared to those other omic layers, which were less. And this really suggests the importance of the metabolome as an omic layer for understanding exposure response. And this isn't surprising since the blood metabolome here, it's the largest ohm depicted, but it's also considered the most interactive with the exposome because xenobiotics can initially interact with endogenous metabolites entering the body. In addition, xenobiotic biotransformations are similar to that of metabolic pathways and sometimes involve the same enzymes. So this relationship is consistent beyond assessing a single individual, like in the previous example. And here, another multi-omic analysis was performed on 1,300 school-aged children and their mothers during pregnancy from 6 European cohorts as part of the Human Early Life Exposome Project. And exposure assessment tools here included mass spectrometry-based measurements of biomarkers of chemical exposure in urine and blood, exposure monitors, remote sensing, and self-reports. And this resulted in 116 environmental exposures measured in childhood. So in these children, they performed in-depth multi-omics molecular phenotyping, including serum metabolites, urinary metabolites, proteomics, gene expression, methylomics, and microRNA expression. So for the childhood exposures, you can see the significant associations depicted here as dots with the various exposures along the x-axis here. Each of the components of the omic layers are distinguished by a color. So the urine metabolomics are the yellow dots and the serum metabolomics are the red dots. And this childhood exposure, the childhood exposome was associated with all of the omic layers. We see all the colors here, but the serum metabolome showed the highest number of associations with 43. You can see that large portion of that, the pie chart. So given that the metabolome measured here was limited to only about 200 serum metabolites and urine metabolites, it's really further evidence that the metabolome in particular is strongly influenced by the immediate environment and therefore particularly sensitive for looking at biological response. So while these studies looked at associations between tens to hundreds of exposures with multiple omic layers, have provided a deeper understanding of how we can capture and measure biologically relevant environmental exposures. These highly characterized studies are just emerging, right? They're limited. And the exposures investigated in these studies are biased by what we can measure, what chemicals we can identify or annotate, and which chemicals we can detect in large enough numbers of our study population to be able to perform statistical analysis. And because of these challenges, the concept of the functional exposome has recently emerged. And the functional exposome has an emphasis on sort of characterizing that space within the exposome paradigm where exposure is meeting biology. And this approach focuses on biologically active sites or pathways and the classes, subgroups, or mixtures of chemicals that interact with these sites or pathways. And so I consider these biology-driven approaches sort of like an exposome dimension reduction technique, really, to help us overcome some of these challenges with low-dose detection, unknown annotation, and complex mixtures. So there's several different approaches to this, and some are effect-directed analysis, adaptomics, and others. And here I'm going to focus on metabolomics as a biology-driven approach. And this is because metabolomics is a tool for focusing on molecular interactions between an exposure and biological response, and measured as alterations in endogenous metabolic pathways. And so in this recent review of metabolomic studies of PFAS in humans, a few pathways and metabolites were reproduced across several analyses. And most of the 11 studies were cross-sectional. They were measuring metabolome and PFAS in the same sample. But despite these differences between study designs, the sample types, the data acquisition and processing methods, and even the annotation confidence, 8 studies reported significant associations between 4 dominant types of PFAS or PFAS mixtures and metabolites. So a total of 6 studies reported associations between PFAS and amino acid-related metabolites, and on the pathway level, reported amino acid metabolism and lipid metabolism being some of the most commonly associated pathways with PFAS exposure. So given the variability between the studies performed on so many different levels, and seeing some of these overlap in the findings is very promising. This is far from conclusive, but they suggest that PFAS and PFOA in particular are biologically active in the metabolome. And since recent regulations support the science that PFAS and PFOA are linked with adverse health outcomes, these results point to specific pathways that can be focused on for future studies. And so here's another example that focuses on identifying lipids as active metabolites. These researchers built a database of xenobiotics, which they screened in untargeted data from 120 serum samples to look for associations between these chemicals and a list of 9 targeted measures of lipids. And this table font is quite small, but I've highlighted in yellow where about half of the significant associations between the chemical suspects and the targeted lipids were with lysophosphatidylethanolamine 18-0. And even looking at only 9 lipid metabolites, these findings support the idea that there are key active metabolites measured in the metabolome that may be acted on synergistically or antagonistically by mixtures of exposures. So to give an example from my own work, we are trying to address this using metabolomics and a dimension reduction technique based on the biological importance of reactive metabolites. In this model, we assume that highly correlated metabolites contain more biological information than those metabolites that are isolated. And then we link these functionally important metabolites to exposures and health outcomes. So in this workflow, we first determine the correlated clusters. And here as examples, you see 3 individual clusters, right? Correlated features. You see a red cluster and a gray cluster and a blue cluster. So step 2 is looking for associations with the health outcome. And here it's sort of depicted as a triangle. And looking for associations between these health outcomes and these correlated clusters. And in this example, the health outcome is only associated with a feature from the blue cluster and the red cluster. So the gray cluster is removed for further downstream analysis. And step 3 is combining these into a new network map that now only includes the correlated clusters where at least one feature from that cluster is associated with the health outcome. And then finally, step 4 is now including these exposures represented by squares that are associated with the metabolite features. And so we first looked from the perspective of identifying active metabolites that were active to exposures. This was done in a study of 180 adolescent girls. And we narrowed down our metabolome clusters that were correlated and associated with targeted trace elements in PFAS, and then look for associations between these features and puberty outcomes. The first thing we noticed was that manganese was associated with the metabolome more than any other trace elements that we had included. And this was surprising since there's very few metabolomic studies of manganese in the literature. And so this could be pinpointing some new potential avenues of research. In addition, while there's very few direct associations between these environmental chemicals and our puberty outcomes of BMI and age at menarche, we found that there were several exposure active metabolites that linked exposure biomarkers to our health outcomes, even when there were no direct associations. So for example, here, androsterone sulfate was positively associated with BMI percentage and negatively associated with PFOA, which may be highlighting endocrine disrupting properties of these chemicals. So since metabolomics is really at that interface where exposures meet biology, we could actually flip the workflow around. And here we wanted to generate hypotheses on important pathways that may be explaining observations in the literature between nutritional factors and breast cancer risk. And here we first narrowed down our metabolome clusters that were correlated and associated with breast cancer. So this time looking from the health outcome side. And then look for associations between those breast cancer active metabolites and nutrition and lifestyle factors, including physical activity, fruits and vegetables, supplements, meat, tobacco, and alcohol use. And here you can see how the lifestyle factors connected between these correlated feature clusters to form a single network. And that alcohol, folate, beta carotene, and meat were the most central, with B12 the most adjacent, suggesting that these central ones have a larger influence on this breast cancer active metabolome. And the other lifestyle factors were not associated and dropped out. So we then annotated some of these features, and our preliminary results show a cluster of bile acids up here, some phospholipids, and a chemical that had MSMS data consistent with trazodone hydrochloride, which is an antidepressant, as well as 13-HPODE, which has been linked with inflammation in the literature, suggesting a potential pathway linking these lifestyle factors to breast cancer. So to summarize, I hope that I demonstrated that the metabolome and the exposome can inform one another. Although it's still an emerging field, metabolomics has been demonstrated as a powerful dimension reduction technique of the functional exposome. And sort of new developments in this area will continue to address challenges with mixtures and low concentrations. I also want to bring up some challenges. While it's difficult to standardize the untargeted metabolomics workflows between research labs due to various differences in analytical platforms and bioinformatics used, transparency in providing the details of these methods and how annotation was performed, and providing those in the publications, providing reproducible code for the data processing will really help with interpreting metabolomics associations across studies. And so I do want to acknowledge collaborators on the studies I presented, my lab team, and of course, funding support as well. So thank you very much, and I'm happy to answer some questions.
Fabulous presentation, Lauren. Lots of food for thought here. You presented a lot, and I'm just trying to wrap my head around this. So the PFAS issue, such a massive class of chemicals, and I think we're all trying to understand, what effect they may or may not have. When I was listening to your presentation, one of the things I'd wondered about is, in your analyses, can you start to do things like filter based on the structure? So maybe it's the chain length of the PFAS as well as those functional groups. So maybe by, sulfur, sulfonates, those kinds of things, to see whether or not you sharpen the associations. I wonder if you could comment on that.
So in terms of pulling those out of the data, we can specifically screen the untargeted data for chemicals from certain classes. For example, we can do this with halogenated compounds using things like mass defect and other sort of techniques that relate to the chemical structures. We can also filter by fragment losses of a molecule like a sulfate or a glucuronide to show that they're biotransformations. So we can start to pull those out of the data on an individual basis. I think one of the questions that, when we start to pull these out, we can see that there are these sort of scattered exposures of specific PFAS chemicals that we can even, there's now amazing libraries that we can purchase that'll help us have really confident levels of annotation for them. But we're starting to see them sort of scattered across some of these, the individual samples. And so how we address that statistically, when you have a lot of missing values, I think is still something, coming from that side, is hard to do from the individual PFAS side, right? So starting to think about ways that we might be able to do that, looking at specific biological interactions where we might see them from pathway levels where multiple from different functionalities may be affecting that specific class, that specific pathway might be a way to start to get around that, or some way of combining them biologically.
Okay. Thank you. So the next question is a two-part question. So you talked about signals versus metabolites, unknown and annotated. And I was wondering if you could tell us a little bit about two things. So first is the differences between these and how they relate to the workflows that you described and the transparency that you just mentioned, because you hit on such an important aspect. Using different platforms, it's really hard to compare data because a lot of the analysis tools, for example, are proprietary. So that's one thing. And then the other thing I wanted to add on to that, you showed a really nice graph where you were showing the relationship between how well metabolites were tracking with exposures. And I'd wondered, what was also showing up, not as strong, so I think that was maybe a 40%, but you also, maybe 20-ish% was the microbiome. And so what I'm wondering is, are you, do you think that the microbiome is playing a bigger role, and it's just a matter of getting your, being able to actually tackle it? Because I do wonder if some of those metabolites are actually coming from the microbiome, and maybe even as a function of time. So taking several samples across time, because what might need to happen is that microbiome gets reestablished and changes up. So I wonder if you can comment on all of that.
So I'm going to try to start at the beginning, and I hope I don't forget along the way all the questions, but these are great questions. So first, because in a targeted assay, it's going to be a metabolite. You know what it is, you're looking for it, you're sure it's a metabolite, and so you can call it a metabolite. When we do our broad screening, the signals that come directly from the instrument, they're features. We don't know what they are yet, and until they're given an annotation, we designate them as a feature. And because that process of identifying what those metabolites are can be very time consuming to do it rigorously, it's usually left to a later step. So we call them features until we narrow them down into what we think is important for the study. And then we can dive in and start to do some of these annotations. So that's sort of how the workflows relate and to that description. What was the second part? Do you remember? We could talk about transparency. Yes. Okay. So transparency. Right?
Transparency is fine, and then the microbiome piece.
Yeah. So and so how this relates to transparency, which I think is also important, is because there are, there's the analytical methodology which can be described and can be easily reproduced, but all of those parts from identifying those metabolites, whether you're using an in-house library, you're using available databases, analytical standards, automated softwares. There's so many routes, and a lot of it are manual, which makes it hard to make it reproducible. So I think documenting is really important so that the level of that, all the evidence that has been used to come to that conclusion of the annotation is available. And so there's been lots of confidence type of scales that have been developed and are for the community are reinforced that they should be included with those publications so that people are clear about how confident these metabolite annotations are. But it also comes down to the code, right? From, you have these 20,000 signals that you filter down to a set of 20 that might be associated with your health outcomes. And all of those steps from, filtering to adjusting and then the actual data analysis needs to be reported and provided as code so that people can try to reproduce this data and compare across studies. So I think that these are important aspects that need to continue to be used well within the community. So I guess the final question related to the microbiome, I think this is really interesting, right? One of the first biggest studies that came out related to metabolomics came out of Stanley Hazen's group 10 years ago, maybe more at this point. And it really showed that there was this relationship between carnitine and the microbiome that was linked to cardiovascular disease. And all of these studies, there was multiple studies that came after it with mouse models and really providing causality between the metabolome, the microbiome, and this health outcome. So that was sort of the first study. And I think this is coming out more and more that we're seeing these relationships between the microbiome, which obviously has an effect on the metabolome. And they're coming out now. And I don't know that we have a full grip on separating them and how much is the overlap, but these sort of multi-omics assays, whether we're doing epigenetics and metabolomics, whether we're doing microbiome and metabolomics, I think are really going to be helpful in trying to tease out those relationships. But I think they're all interrelated. And so we, as much as we can start thinking about them as layers that are connected in time, right?
Yeah, I like what you said, as layers connected together in time. I think that's really key. So when Andrea gave us his presentation, he had this long-term, what could we have a dashboard that we could sort of let people know, are you good, are you bad, with being exposed to one thing or another. So with that in mind, I'm kind of wondering, from your perspective, do you see metabolome analysis being used clinically one day as a way to understand toxicant exposure and maybe identifying those who might be at higher risk for a given disease, those kinds of things?
Yeah, so I mean, there's an entire subfield of clinical metabolomics, right, that are working on doing this, really trying to, come from huge databases of what is a healthy metabolome, right? And that's the starting point. How do we find healthy versus not healthy, right? And that within itself is a huge endeavor. I think there's a lot of very interesting studies that are coming out about the role of different environmental exposures in terms of, food additives and obesity, and I think propionate is one of them, with, a food additive and obesity and type 2 diabetes. There's also been some interesting studies that have shown that genistein, which is from soy foods, has some adverse interactions with anti-cancer drugs. So I think as some of these studies are coming out, there's going to be more and more interest in understanding how these environmental factors are going to really relate in a clinical perspective. I think we have the same vision that, in 10 years, there will be something, and I don't know how long it's going to take. But I know that there is a webinar scheduled in this series to talk about precision exposomics. So I'm looking forward to hearing that, to hear more about it.
All right, thank you so much, Lauren. And Andrea, I do apologize. I had a question ready for you, and I'm afraid we're going to have to do that by email because we're past time. This has been a lot of fun. Thank you very much for taking the time.